Andrew Stout from Tufts University. Well known in this community. He has been attending this conference for quite some time. He is today going to talk about simple serum-free media for bovine satellite cell culture. Andrew, the floor is yours.
Okay, excellent. Thank you so much, Mark, and for having the conference virtually. I was devastated when I thought that it wasn't going to happen at all. So I'm really happy that it is. So as Mark said, I'm going to be speaking about a simple serum-free media for bovine satellite cell culture. And to kind of frame this, I think that if we think of Professor Cosgrove's talk as kind of a novel of scientific production, I like to view this as somewhere between sort of a short story and a haiku. So it's a pretty kind of simple story that I want to tell everybody, and I hope that people can take it and go forth with it. So when we think about serum, I think that the field of cultured meat has a big kind of awkward relationship with serum because we use it a lot, and I think that it really complicates things. And so to kind of break this relationship down, I think that it's important to realize that serum-free media exists for muscle cell culture. I think that when we talk to people outside of the field, and when sort of the media and the public talks about the challenge of serum-free media, it's often missed that we have options for serum-free muscle cell culture. They exist, they're characterized in literature, they're purchasable, etc. They are not used ubiquitously in cultured meat research work. And does that reduce the relevance of that work? It can, I think. I think a lot of times, you know, serum is kind of this caveat that we have to put on every single communication we put out there, where we say, you know, this would obviously be important to understand in a serum-free system, and the results might change a little bit in a serum-free system, but we think that they're still relevant for X, Y, and Z reasons. And I think needing to use those caveats is a challenge, particularly just in our communication of the research that we do. So it kind of begs the question, you know, if we have serum-free media that we can use, and if using serum is sort of this big communication challenge for the field, why do we still use it? And I think that simplicity is one big reason. I think that, you know, characterization of serum in media and knowing that serum will work in your cell cultures is very clear. And so we often, you know, if the primary thesis of the work or the primary hypothesis of the work isn't about media, then it makes the most sense and it's simpler to just kind of toss in serum. And another reason is that a lot of these serum-free options that are out there aren't perfect. So when we think about serum-free media for cultured meat, I'd like to sort of posit that simplicity is valuable from a cost perspective. Food safety is essential from a regulatory perspective. Animal-free nature is essential just kind of from the core tenet of cultured meat research. An open recipe, I think, is really valuable for particularly academic research in the field. And then efficacy compared to serum is another big factor because a lot of papers that have presented serum-free media for muscle cell culture in the past, they don't actually compare it with serum in terms of growth rate, et cetera, because just kind of the priorities in sort of medically aligned muscle tissue research are different than the priorities in food aligned muscle tissue research. And so basically, this is just sort of a slide that I hope captures the fact that, you know, we don't have a serum-free media that checks all of these boxes yet. So what we did for this haiku, I guess this was born sort of a Friday afternoon project. And basically the idea was that we were looking for something simple, something affordable that could be a tool for researchers and could be a starting point for optimization. So in this project, we weren't trying to produce the greatest serum-free media in the world. We weren't trying to hyper optimize anything. We kind of just want something that's accessible to all of y'all doing your research and with which you can run on your now caveat-free experiments or that you can further optimize. And we were inspired by this paper that came out recently about a low-cost serum-free medium for iPSCs. It came out of Paul Burridge's lab at Northwestern, and it was a really, really cool paper because basically they showed that they could engineer growth factors for high stability and low cost production. And they could achieve just kind of weekend-free, easy to use, low cost culture of iPSCs. And kind of since that paper came out, a company has been formed called Bovine Bioscience based on that paper, and they're producing basically aliquots of these supplements that you can order and just add in to a basal media. And so in the paper, when they engineered these growth factors, they were able to get the cost down to $16 per liter for this B8 media. So we kind of thought, oh, this seems like a really great starting point. Let's just kind of see how it does. So we took some bovine satellite cells that we have in the lab. These were isolated from a 1-month-old cow, and we've characterized them in the past for PAX7 expression. So looking back at Dr. Cosgrove's talk, this marker of satellite cell identity and muscle stemness. And then we've carried them out through differentiation processes and shown differentiation. And what we did for this work was it was really simple. We basically seeded these cells on laminate-coated plates and cultured them in B8 and did DNA quantification to kind of track our cell number after 3 or 4 days of growth. We stopped at 3 or 4 days of growth because the cells got confluent by day 5. And so that's kind of where we are now. A caveat that I have to add is to say that short-term results in this work does not necessarily equal long-term success. And so for this project, we're in the process of working on our long-term studies, but the data I have for you today is short-term studies. So I think it's still valuable data, but please accept that this could change in the long term, and there could be tweaking and optimization that we need to do moving forward. So the different media that we explored, just to kind of go through this. So our normal growth media is DMEM with 20% FBS and 1 nanogram per mil of fibroblast growth factor. And just, quick Googling of supplier pages, it looks like I can buy that for about $125 a liter, all those components. Producing my own B8 using non-engineered growth factors and just store-bought components that I try to buy in as big of bulk as possible, I can make B8 for about $75 a liter. And then high def, ordered from Define Bioscience, is this same basal media and basically adding in one of their aliquots, which they provided some for us to use. And so they're currently on pre-order for $125 per liter. But I'm sure that, considering this reduced cost that was shown in the paper, that will potentially be brought down in the future. So to start, we just looked at cell growth in growth media, in 100% B8 or in ratios of the two. And kind of the takeaways here, looking at B8 and at the high def B8, are that with B8, we have cell growth. You can see the cells in these microscopy images at the bottom. But our cell growth is less than in our serum containing media. Ratios of the two have higher growth, which I'm happy to discuss later, but I won't really get into right now. But kind of the core takeaway is that these media seem, they indicate that they support proliferation. Growth over 3 days is reduced compared to FBS containing media. And when we just look at the cells, they look pretty happy and healthy, just kind of at a quick glance. So what we did after that is we did a little bit of optimization. So the first question we asked, can we lower the cost of this even further? And largely, can we do that by reducing the amount of FGF that's in this media? And can we also potentially eliminate these other growth factors? FGF is by far the most expensive part of B8, just because it's the growth factor that's present at the highest levels. And so when we basically do this same experiment, a short-term growth experiment, looking at dilutions of FGF out of our system, we can see that down to, so B8 is 40 nanograms per mil, down to 5 nanograms per mil, we don't see any significant change in growth. So that indicates that we might be able to, at least in the short term, reduce our FGF concentration and still have good growth, therefore reducing the cost. And it also looks like we can potentially eliminate TGF and NRG from the systems. Again, caveat at short term, but kind of just something to build off of. So the next thing we did to optimize was we saw whether we could add supplements for increased growth to try to get that growth level up to where it is for FBS. And so we tried a lot of supplements that didn't work. We tried a range of growth factors. We tried a range of steroids. We tried a range of small molecules. We didn't see any changes in short-term cell growth in the concentrations that we explored, the formats that we explored, etc. That's not to say that we should totally write these supplements off. There have been papers that have shown success with these different factors in increasing proliferation of satellite cells. We just didn't see it kind of in the formulation that we were testing and exploring. However, we did find some factors that did have significant effects. And so to kind of step through these, we saw that some pro-inflammatory and some anti-inflammatory factors significantly increased cell proliferation in the short term. So basically here on the left with zero supplement, you can see all of this data is just relative to that standard B8 zero supplement data. And so you can see that with low levels of interleukin-6 and low levels of curcumin, we saw increased growth. We also saw increased growth with the inclusion of fatty acids like linoleic and oleic acid. And there's a lot of evidence, literature evidence to support that those would likely help in this situation. And then really most importantly, we saw a lot of improvement with the inclusion of recombinant albumin, which is a signaling and carrying protein. And so what we did after this, and this was really exciting, but after this, we basically wanted to just do a quick look at the combinations of these factors. So when we just mix the factors in different combinations, how has growth changed? And the main things that I'd like you to look at here are where the red arrows are. So here is IL-6, here's albumin, curcumin, fatty acids, and then these are just the combinations of those factors. And basically, you can see that when albumin is present, it vastly improves growth compared to B8, and it allows growth that's comparable to our serum-containing media over this 4-day period. And pretty interestingly, I think that the positive impact of albumin seems to kind of just overtake any of the positive impacts of these other factors. And so this is, I think, not to say that, I think that two things could be going on here. One could be that albumin is just, compared to albumin, these other factors just become sort of noise, and it's such low level changes over this short time span that we're not seeing it. Or there could be something going on biologically here where the combination of factors have reduced growth. I think that, I'm not willing to sort of stake my name on biological things here, just because there's a degree to which this is a relatively coarse assay over a short time span. But kind of the takeaway is that, it looks like we have an option that not only works really well, but is also very simple. And that's just the inclusion of albumin in our B8 formulation. And so, like I said, the kind of next steps that we're looking at are long-term multi-passage growth analyses of these media. And then just to kind of look back at this cost consideration. So we're doing these long-term growth experiments with our FBS containing media, our homemade B8, our homemade B8 plus recombinant albumin, B6, which is reduced FGF, and then neither NRG or TGF, B6 plus albumin, and then the high-def provided media to us, and high-def plus albumin. And during that process, we're going to do staining for cell phenotype during proliferation and after differentiation. But I hope that the kind of takeaway conclusions here are that I think that this is a really promising starting point for serum-free media. I think that there's a lot of optimization that I'm sure can happen. And I think that there's a lot of people hopefully listening to this who are excited about, performing that optimization. But but kind of from where we stand right now, this looks like a promising option for a research tool for groups to use for an optimization starting point, and hopefully to kind of reduce the need of that caveat where we always say, this would be better without serum, but here are the results anyways. And so with that, I'd like to thank my team, the Kaplan Lab at Tufts University, for all of their help. Particularly John and Addie have been helpful with this project. Thank you to Divine Bioscience for providing those aliquots of their material. Thank you to New Harvest and the Good Food Institute for funding the lab, and obviously Tufts University. And thank you to all of you for listening, and I'll gladly take questions.
Well, Andrew, thanks a lot for a wonderful presentation. I think you need to revise your concept of a haiku. It was slightly longer than that, and slightly more complex as well. There are a few questions. We don't have a lot of time, and I don't want to be too late. One question you kind of alluded to in your kind of future perspectives, whether you have diluted not only FGF2, but also the other growth factors, NGR1 and TGF-beta, but I guess you are going to do that, and you have not done that yet. There's a question about the albumin, which albumin you're using, whether this is the standard fraction 5 or the RIA grade version.
So it's recombinant human albumin. So it's just the albumin protein, not the fraction that also has the fatty acids and other components in it.
Right. And a question that we're always asking if we test media is whether the cells are still able to differentiate after that, or whether it affects differentiation.
Yeah, so that's what we're working on now. I guess the short answer is I don't know. The slightly longer answer is that I feel good about it, even without knowing, because when we were first starting the experiments, we would culture out past that 4-day mark, and we saw that in the B8 cells at day 5 and day 6, the cells were already starting to fuse and differentiate. So I think that indicates that there's at least not an immediate full loss of differentiation capacity. Whether that holds, over the long span, I'm less sure on. But also the media ended up being formulation-wise relatively similar to a media that have been presented in the literature in the past that have shown maintained differentiation capacity.
Right. There are quite a few other questions, so also to you, please look at Slack and try to answer them on Slack.
Yeah, certainly.
Thanks a lot, Andrew.
Thank you.